Hello, everyone. So today we're going to talk about viruses and some other acellular pathogens. And so to start the story out, I want to tell you a little bit about the history of tobacco. So obviously tobacco had been around since before 1847, but in 1847, Philip Morris founded the company, for Philip Morris in London. And in 1875, R.J. Reynolds founds his tobacco company in North Carolina. So here are two of the, largest tobacco producers in the world coming into being in the late 19th century. Around that same time, the first cigarette rolling machine was created. So that happened around, I think 1881 or something. And so that's what we see here is this cigarette rolling machine. With the, advent of this machine, tobacco use became, or cigarette use became more common. And just during this time as a whole, tobacco use was growing. And so it became really important for producers of these tobacco products to, control disease in their crop. And a major disease of tobacco is this tobacco mosaic disease, which we can see here in various forms. And we can see these like mottled leaves, which is like a hallmark of the tobacco mosaic disease. And so there's this Russian botanist / early virologist named Dmitry Ivanovsky, who worked on trying to identify this organism. And so he was thinking about it from the standpoint of Koch's postulates. And so if you remember Koch's postulates, you find a suspected pathogen in diseased animals, but not healthy animals. You're able to culture it from the diseased animal, but not the healthy animal. And then if you give that cultured organism to a healthy animal, they will, get the disease and you'll be able to identify it and culture it there. Well, when Ivanovsky tried to do this with tobacco mosaic disease, he was unable to. So he extracted cells from the TMD infected tobacco, but he wasn't finding any bacteria that were able to grow or that were causing the disease. So around this time, remember, he could look at this through a light microscope. He could try to grow it in culture media, but he was having no luck at culturing the organism causing this disease. However, if he took those extracts and gave them to a healthy plant, those extracts would cause disease. And he put those extracts through a filter that was about 0.1 micron. And even the extracts that went through the filter still caused disease. So 0.1 micron is going to filter out the majority of bacteria. And so at the time, Ivanovsky thought that these were bacteria that were just so small, or bacterial spores that were so small that they could fit through this filter, but that he couldn't see or grow. Now, later on, Martinus Biergernik decided that it couldn't have been a bacteria that was causing TMD, and he named it virus, which was Latin for poison. So he was thinking that maybe it was some sort of chemical that was passing through these filters. Now, of course, we now know that viruses are like these things that we can see, and we're going to talk about today. We can't see them with a light microscope, but we can see them with electron microscopes. And of course, in the 19th century, we did not have electron microscopes. So by the end of this module, you'll be able to identify the parts of a virus, describe different types of viruses, explain the viral life cycle, and explain the immune response to viruses. And so first off, let's just talk about them. I hinted at this a moment ago, but viruses are very small. If we look at this, this graph here, we can see that on this end, we have like, you know, some of the largest cells like human eggs, frog eggs. We've got like plant pollen. And this is like, you know, a frog egg is like a millimeter, right? So these are pretty big. Now, if we come on down, bacteria are much smaller. They're about 1 micrometer, right? So that's like a 1,000 times smaller than a frog egg. And keep in mind that bacteria and mitochondria are about the same size, right? So like our animal and plant cells will actually have mitochondria inside of them because, and the thought is that these mitochondria are actually derived through endosymbiosis. So these used to be bacteria that were incorporated into these, you know, eukaryotic cells through a symbiotic process. That's not what today's lesson's about, but it's a sort of interesting side note. Now, our biggest viruses, like the smallpox virus, are still like probably half the size or more smaller than your normal bacteria. And then our smallest viruses, such as like polio, are going to be nearly 1,000 times smaller than a bacteria. And so we can't see these really small things using a light microscope. Our limit of detection is somewhere around probably about 200 nanometers, give or take, to distinguish two, you know, different objects. And so if something's only like 300 or 400 nanometers across total, then it's going to be pretty hard to see and distinguish what it is on a normal light microscope. Now, viruses are acellular, so they don't have a lot of the things that are necessary to call something a cell, like a plasma membrane. You know, those are the like inner and outer membranes of bacteria. They have no cytoplasm. They don't have any ribosomes, right? So they actually are really, you know, simplistic compared to a cell. Their organization is much simpler. Now, the other thing that's interesting about viruses is that they have very diverse genomes. They can be DNA or RNA. And so if we look in these different example viruses, we see the genome in here. We see a DNA virus here, a DNA virus here, and an RNA virus here, right? Now, they're all going to have some sort of protein coat, which sorry, this is a poorly set up slide, but they have a protein coat, and this protein is called this protein coat is called a capsid, right? So this part of this virus is the capsid. This whole thing here is the capsid, and then inside here, around the genome, is the capsid. So these little circular monomers here. And so every single virus is going to have some sort of genome, DNA or RNA. It's going to have some sort of capsid, like here, here, and inside here. All right. Now, the individual proteins in the capsid are called capsomeres, right? So if we look over here at this one, this like triangular piece is a capsomere. These little ovals are capsomeres, and these little circles are capsomeres. Now, when a capsid has a genome packaged inside of it, that is called a nucleocapsid. So since we have a genome there, that's a nucleocapsid, that's a nucleocapsid, and this little guy in here is a nucleocapsid. Now, some viruses have this lipid layer that surrounds that nucleocapsid, right? So here we see our capsid with our nucleic acid genome in here. But over here, that is actually surrounded by these lipid, this lipid layer. This is called an envelope. And so this is called an enveloped virus. On the other hand, our non-enveloped virus is called a naked virus, right? And so if you just have a nucleocapsid with no lipid layer around it, you're a naked virus. But if you have that lipid layer, you're an enveloped virus. Now, viruses will usually have some sort of spike protein used for attachment. So we see on this naked virus here, we see that these spike proteins are glycoproteins that are actually used to attach to cells. And we can see that in this electron micrograph here. We can see the little spikes coming off. Now here we have HIV, which is an enveloped virus, and the spikes are actually going to be in the envelope. Now keep in mind that generally speaking, the spikes are going to be on the outer layer of the virus, whether it's naked or enveloped. And so it's a little bit harder to see those spikes here, but we can see on these cells, we've got these little protrusions coming off, and those are little virus particles. Now, this to kind of put this in perspective with our current world, the coronavirus vaccine for COVID-19, which carries which is a mRNA vaccine, that mRNA actually encodes for the spike protein of your COVID, of your coronavirus. And so those are these little guys out here. So what happens is when you get that vaccine, the mRNA is turned into proteins by your cells. And when they make those proteins, it actually makes these little spike proteins. And those little spike proteins can trigger an immune response. Now you can have some other specialized stuff, like in the T4 bacteriophage, they have these little tail fibers, right? And so the tail fibers attach to your cell or to a bacterial cell because it's a bacteriophage. Keep that in mind, that bacteriophage only affect bacteria. And something to just kind of like keep in mind in general, and I think we'll talk about this a little bit going forward, is that viruses are generally highly host specific, right? So whereas like a lot of bacteria may cause disease across different types of organisms, like as long as they kind of operate within the same body temperature, viruses are very host specific. So a bacteriophage are only going to affect bacteria, but specifically, they're probably only going to affect one type of bacteria. So like a coliphage is only going to affect like E. coli. A Campylobacter phage is only going to affect Campylobacter. So a quick knowledge check, take a moment, sketch a virus, label the capsimeres, and the genome, and spike proteins. You can make your virus enveloped or non-enveloped, that's up to you. Take a moment, pause the video, and check back in. All right, so hopefully you drew something along these lines. You've got your capsid, which could be any of these examples, right? Your individual proteins are your capsimeres. Inside of the capsid, you should have your genome, which can be RNA or DNA. And then you're going to have spike proteins, right? Now, if you have, for example, the bacteriophage, you wouldn't have spike proteins, but you might have tail fibers, right? So here's spikes, here's spikes. And don't worry about your art level. If you are not an artist like me, we're just looking primarily for you to practice thinking about those different parts of the virus. Now, viruses can be morphologically diverse. You've seen that a little bit with the picture we were looking at before. For example, this TMD or the TMV tobacco mosaic virus is this helical structure, right? And so the genome's actually packaged inside of this. This would be your capsid. Then we have the icosahedral. This is rhinovirus, which causes like a cold, right? And so it has like these 20 sides to it, or these pretty cool looking, it's a pretty cool looking shape. So it's similar to what you see with that bacteriophage here. This is maybe a little bit easier to see. This is icosahedral. And then you have these complex viruses like this variola virus here. And so you can see that the capsid is inside of this envelope here. Now, viruses are obligate intracellular parasites, and they can infect all sorts of organisms. For example, this is a bacteriophage, which we looked at before. It's got some specialized parts on it that we don't need to worry too much about. But they can also infect plants. So this, we talked about tobacco mosaic virus. We also have the cauliflower mosaic virus. So we've got these little pieces here are your virus particles, and you can see that it causes this mottling of the leaves. And viruses infect animals, right? So here's a nice complex virus that causes, or this is coronavirus, right? So we've got our envelope on the outside, and we've got our capsid on the inside, packaging our genome. Viruses also have very diverse genomes. So we're not going to worry too much about the differences between how these things work, but I do want to point out that they can come in multiple flavors, right? And this doesn't work with our general thought of how, like how we think of proteins being made in molecular biology, right? Which is generally going to be DNA to RNA to protein. But in viruses, your starting material doesn't always have to be DNA. It can be. We can have double-stranded DNA viruses, right? Like these. We can also have single-stranded DNA viruses like this. So all organisms, all living organisms, bacteria up to all eukaryotes, all prokaryotes have a double-stranded genome, a double-stranded DNA genome. But viruses can have single-stranded DNA, right? Viruses can also have single-stranded RNA, and then viruses can have double-stranded RNA. And then we can have retroviruses, which actually go from an RNA back to DNA. So that's something that doesn't happen in like eukaryotes or prokaryotes either, right? So it's usually, or it's always DNA to RNA to protein. And so here we can just do RNA to protein. We can also do RNA to DNA to RNA to protein. I don't need you to memorize all that. I just want you to recognize that your genome can be DNA or RNA, right? And they can be diverse in how that DNA or RNA exists. And interestingly, since we don't produce double-stranded RNA in our own bodies, double-stranded RNA is a big indicator that you have a viral infection. And so your body will detect this double-stranded RNA and begin immune response if your virus has that. Now, the other point of clarification here is not every virus can do all of these things. Each virus has its own type of genome. For example, HIV is a retrovirus, right? And coronavirus, I believe, is a single-stranded RNA virus. So, there are these differences between viruses that exist. You don't need to worry about identifying exactly what virus has what, but just know that different viruses have different genomes. So take a moment, think about these two questions. What type of virus attacks bacteria, and are they harmful to humans? Pause the video and come back once you've answered these. All right, so your viruses that affect bacteria are called bacteriophages, and they are not harmful to humans. They can only affect bacteria. In fact, there are some places that are studying the effects of phage therapy, which essentially tries to use bacteriophage to fight off infection. And this is a possible alternative to antibiotics that is being explored. Now, Viral life cycles can vary a little bit, and so we'll talk about both the lytic and lysogenic cycle here. And so in the lytic cycle, the first thing that's going to happen is that the virus is going to attach to the host, right? So up here we have a bacteriophage attaching to our bacteria. And then in this case, it just inserts its genome, right? So what this bacteriophage does is it just basically sends its genome from the capsid through this sheath into the cell. Sometimes it's just the genome, but sometimes it's also the entire virus particle, right? So here we see an envelope virus that's being endocytosed into your cell. And then once in the cell, the capsid is actually released. And here the genome will be released from the capsid. Now, this viral genome that gets inserted is going to hijack host enzymes to replicate using host proteins, right? So it's not, that's kind of a poorly written sentence there. It's not replicating the host proteins. It's using host enzymes to make proteins for itself, right? So these are going to be viral proteins made by the host. And that's what we see down here, right? We've got capsids, we've got other proteins, and then even the genome is going to be replicated as well. And once the viral proteins are assembled and the genomes are replicated, they're going to be packaged together, right? So these genomes are going to be put inside of your capsid and the rest of the virus is going to be assembled. And then you get viral release through lysis, right? So these cells are going to, or these viruses are going to lyse this cell. Now, lysogenic viruses attach and enter the cell similar to the lytic phages, right? So you're going to attach, you're going to insert your genome. But instead of immediately going into replication, you're going to have some intermediate steps. So what happens is we can see this here coming into the cell in this first image. And in the lysogenic stage, the actual viral DNA gets inserted into your host DNA. And while in the host genome, as the host cell divides, then the viral genome also divides. So we see that that makes it into 2 cells now instead of just 1. And this will go on as long as cells divide. Now that when that bacteria, like in this case, if it's a bacteriophage, the genetic material, when inserted in the host, is called a prophage. So that's just a term you may see. Now, under stressful conditions, the viral genome is excised, so it's removed. And then it basically just enters into the lytic cycle, right? So you're going to hijack the host machinery to replicate the genome and make new virus particles, eventually leading to lysis. Now, the herpes viruses are known for doing this. And so think about stuff like cold sores or shingles. Shingles is a form of the varicella zoster, which is a herpes virus that causes chickenpox. You can see here it's radiating along the trunk, along the nerves. So the varicella zoster kind of inserts itself in nerve cells during lysogeny. And here we see a cold sore on this person's lip, right? And that, again, most of the time, this occurs due to some sort of stress. So take a moment, explain the life cycle of a virus, like the herpes virus, from lysogeny to the lytic cycle. So hopefully you're able to go through this process. I'm not going to go all the way through it again, but just it should include attachment, insertion of DNA into the genome, replication with the host, excising, and then explaining the lytic cycle, which would be assembly or replication of genome and proteins, assembly, and lysis. So viral detection. So as I mentioned before, nucleic acids, especially double-stranded RNA, and as well as proteins, which are not nucleic acids, they're a separate type of molecule. But that's how we're going to detect viral infection, right? So oftentimes it'll be like those spike proteins, which are going to trigger an immune response, or something like double-stranded RNA, which will trigger an immune response. Now, when we think about how the cell reacts, we produce interferon when we get infected by a virus, right? And so here we can see interferon being produced. Here we see our viral particles. Now, interferon is going to do a couple of things. It's going to turn down protein synthesis, right? So the reason we turn down protein synthesis is, remember, the virus is going to basically hijack our protein synthesis machinery. So we don't want to be making those proteins for the virus. Now, interferon is also going to signal nearby cells to undergo apoptosis, right? Some people call it apoptosis, but it's the same thing. Apoptosis occurs, basically it's programmed cell death. And so interferon is going to tell neighboring cells to basically, completely shut down. And interferon is going to trigger immune cells like this, what I'm guessing is a macrophage here, to come and basically try to find and eat infected cells. And eventually, you can get a specific immune response. Antibodies will be produced by B cells to neutralize those viral proteins, and hopefully you can get immunity. Now, that just as a thing to make clear, is these interferon that's being produced, which can come in various forms, is a type of protein. So what is the primary protein produced after a cell is infected by a virus? Take a moment and answer that question. All right, hopefully you came up with interferon. There are a couple of different varieties, but that is the primary type of protein produced to virus infections. And so with that, hopefully you have a better understanding about viruses, and take some time and check out the next lecture, which will be a very short lecture on some virus-like acellular particles and some other acellular infectious agents. Thank you very much.